To all the participants, I'm here to give you a talk on the topic diversification of freshwater algae in lentic water bodies of Telangana state. And the topic I have chosen is the work compiled by our scholars who have worked in our department. So the experimental work carried out by all the scholars is compiled, and I'm here to present the work. And before I exactly go into the work, what we have done, or before exactly what I'm going to present, the experimental analysis, before that, I would like to on aquatic ecosystem, because the topic we have chosen is the work carried out in lentic water bodies. Any of the community of the organisms which are in a water body dependent on one another, and their environment constitute for aquatic ecosystem. And these aquatic ecosystem is again categorized into types like freshwater, wetlands, estuaries, and oceanic. So freshwater ecosystem is again categorized as lentic and lotic, which is to be shown in the next slide. And coming to wetlands, wetlands are the areas where water is present in the soil, and in and around the areas of the soil throughout the year of growing season. So that is water availability within the soil is called as wetland. Coastal wetlands and inland non-peak coming from river. This is how types of axis, but I just started with this. And functional changes are brought forth according to the action and reaction of biotic and abiotic components and its environment functioning constituting ecological. Any ecosystem is dynamic and it with all other organisms to be in balance, that is equilibrium, should be maintained for that them disconnected. Next slide was the we will not admit. He will admit. So is there any need to conserve the biological diversity? This is the question of the hour, where each and every scientist and each and every researcher should understand and answer to this question. Yes, need to conserve biological diversity. We living in India are happy to have rich diversity, and India has got varied inland water resources, and we also have got all water bodies like lakes or rivers, streams, pools, ponds, enriched with algal biodiversity. And the country is phytogeographical and acro ecological diversity endorses the wide variety of agroclimatic zones that harbor a rich repository of biological resource. And freshwater ecosystem comprises environment exhibiting enormous diversity in terms of nutrition, physiological, and biochemical characteristics. Video raatledamma. And algae also play a crucial role of the biosphere, as they can catalyze the process sustaining all life on Earth and act as engines for biogeochemical cycles. And actually, there is a growing realization throughout the world about the urgent need to conserve the biological diversity. However, algae are unnoticed part of the Earth biota and are abundant in freshwater ecosystems, and where they act as primary producers. So as India is a rich country, rich in biological diversity, rich in algal biodiversity, it is need of the art to conserve and to explore all different species of algae, which are abundant in all water bodies, including rivers, lakes, streams, and so on. So it is the responsibility of a researcher, responsibility of a scientist, responsibility of an algalologist to have interest in doing research in this field of hydrobiology or algaology, and also go for further analysis of advanced techniques like algae biology. Sidhu? Huh? Yeah, next slide. Yes. Doing work in part of Telangana, I would like to give you a few lines about topographic features of Telangana. The state of Telangana lies between 18.11 degrees north latitude and 79.0.
Share screen is lost.
So what we need to do
Sir?
Share screen sharing is lost.
the basic and application part.
Screen వస్తుంది.
Answer the question.
Okay, now
biology.
it is okay.
What is biology? Simple.
Please
We are
continue,
not
ma'am.
talking about
You
any
unmute
subject.
yourself, ma'am.
Simple biology. Can anybody unmute themselves and talk?
Yes, sir.
So that
It is
makes
fine
sure
now.
that everybody is awakened.
Yes, sir.
Then
So
the
all these
people
slides earlier were clear, sir.
awakened.
Shall I continue?
Yeah, you continue, ma'am.
So any participant
Yes.
can.
So as we are continuing our research in part of Telangana, I would like to give few lines about topography of Telangana. The state of Telangana lies between 18.11 degrees north latitude and 79.01 degrees east longitude. And geographically, Telangana state lies in southern part of India, and it's bounded by states of Maharashtra to the north and northwest, and Chhattisgarh to the north, and Karnataka to the west, and AP to the east and south. And main freshwater resources are rivers like Godavari, Krishna, Pranahita, and other water tanks. Understanding lakes in Telangana, and what is it that we are benefited out of having lakes in Telangana? This is the answer to be, this is a question to be answered. And most of us in Telangana depend on agriculture. And for agriculture to have yield, you need to have proper irrigation sources. And that is how till now we have got in Telangana around 46,531 freshwater sources. And apart from this, 27,318 freshwater lakes are used as minor irrigation for agriculture and domestic purpose. And people of Telangana are highly dependent on these lakes. And they depend on water supplied from these lakes as irrigation for them to have proper yield from their agricultural farms. And topography and other climatic conditions are also suitable for having this minor irrigation sources. And conservation of lakes is an age-old activity since pre-Satavahana era. And during Kakatiya era, the construction of tanks was carried out with utmost technical expertise. Other than the naturally occurring lakes, some of these lakes were also built and constructed by kingdoms in the past eras. And few of the lakes like Ramappa, Pakal, Laknavaram, and Ganapuram and Bayaram were built by Kakatiyas, which resemble seas and are greatly helped in agriculture. And overall development and prosperity goes to Kakatiya kingdom. And this is how you need to understand the significance of lakes and how we depend on water resources of these water bodies is important. In the title itself, we have taken diversity of freshwater algae, that's phytoplankton. So as you're all aware of phytoplankton and zooplankton, which occur in water bodies. Now I would like to tell you a little bit about phytoplankton. So the term phytoplankton is derived from a Greek word phyton, which means plant, and plankton means wanderer. Wanderer in the sense it should be present everywhere. And of course, phytoplankton is ubiquitous, cosmopolitan, which occur in surface water of rivers, lakes, oceans, but influenced under the direction of wind and current. And they are in diverse habitats, and the composition, distribution, diversity, and abundance of phytoplankton are dependent on some factors like physical, chemical, and biological properties of water, as water is elixir element for metabolic activity or reactivity of any organisms present in the water body. And phytoplanktons are autotrophic, photosynthetic, that is, they can fix solar energy by photosynthesis using carbon dioxide, nutrients, and trace elements. And this is all possible because of the presence of chief pigments like chlorophylls. Phytoplanktons will also act as primary producers and constitute the first level in aquatic food chain. And they occupy enormous range of ecological conditions from lakes and rivers to acidic peat swamps, damp soil, wetlands, snow and ice. And they play an ecological role and are being extensively used as indicators of water pollution because they are natural inhabitants of water. And they also regulate the environment, which are based upon the recycling of nutrients. And they also provide in the environment. So this is about the importance of phytoplankton and phytoplankton. So as an algae, and from among these, we find members of chlorophyll and members of major component of phytoplankton, diatoms, blue-green algae of Sanophyceae, And members of these blue-green algae are abundant in freshwater ponds, lakes, and springs. And they are of a nature of prokaryotic cell, and they tend to concentrate towards the surface waters. And blue-green algae also play previous one. Yeah, coming to global and national status of phytoplankton. Yes. Contributed to your slide share. And it is approximately recognized species. And one more, so that is identification algal. India is one of the most plankton that of in skeletal the composite of phytoplankton by identifying understand for of member of algae. In seed banks, as well as to preserve natural communities by protecting sites of phycological importance. And as we say, these phytoplankton will act as the primary producers in any of the food chain. So this is how we can show. So phytoplankton is consumed by zooplankton. Zooplankton consumed by small fish, and small fish consumed by large fish. So eating an organism and being eaten is called as a food chain, and interlocking pattern of food chain is called as see to the trophic levels of an ecosystem, and also construct a pyramid of energy flow. And this is how the phytoplankton will act as producers in any of the aquatic food chain. And absence of this phytoplankton will act as source of food for other organisms living. So based on these hello, sir. Hello, sir.
Ma'am, your screen is lost. Share screen is lost.
Yeah, it's like connectivity is
First of all, we
not
need to understand that.
proper. Is it from my side?
Because
Yeah,
if we
now
don't
it is coming
understand
again.
What is inside the cell, we cannot change
Now it is
what
okay.
there in the cell.
So based on the points which I have discussed little bit about lentic water bodies and phytoplankton and global and national status of phytoplankton, we have gone for investigations from our own department where we have conducted experiments, and this is how the experiment is. So we have gone for survey of lakes in districts of Nizamabad and Siddipet and Bhadradri district of Telangana. So we have not gone on survey of lakes of total part of Telangana, but part of Telangana state has been covered. And based on that, we wanted to go for exploring the algae from each of the lake and understand for diversity of phytoplankton. And then it was correlated to physico-chemical parameters. And we have taken 6 lakes in Telangana. And as such, these are Komacheru Lake and Erracheru Lake, which are from Siddipet region, and Singagupalam Lake and Karukonda Ramavaram Lake, which are from Kottagudem area of Bhadradri district, and Ashok Sagar Lake and Ali Sagar Lake, which are from our Nizamabad district. So we have taken these lakes and then we have gone for conducting some experiments. So this is a satellite view of lakes: Karukonda Lake, Erracheru Lake, Singagupalam Lake, Komacheru Lake, Ashok Sagar Lake, and Ali Sagar Lake. So seeing to this satellite view, we can go for featuring the geographical characters, and also we can catch up the spatial location of each lake where it is situated. So this is about all lakes where you can see for overview. And we have got Ashok Sagar Lake and Ali Sagar Lake from Nizamabad district. They act as a rich source of water bodies, mainly for irrigation purpose and also for drinking purpose to people in and around living in Nizamabad district. So some of the physico-chemical parameters have to be assessed. So we have taken some physical characters like transparency, turbidity, color, odor, temperature, total suspended solids, total dissolved solids. And diversity of phytoplankton is usually based on spatial and physical factors such as light, temperature, and availability of nutrients. And some of the chemical factors which had to be analyzed are pH, dissolved oxygen, BOD, that is biological oxygen demand, COD, chemical oxygen demand, and chlorides estimation, and estimation of phosphates, estimation of silicates, and total hardness. And what is the methodology we have followed? So we have to see to the methodology which is adopted. And APHA, it's the standard method for analysis of physico-chemical parameters. That is American Public Health Association of all lakes at monthly intervals. And east and west and north and south, we have one site 2, S2, site 3, S3, and site 4 at monthly intervals from all different sites. And all collected sample brought to the laboratory for collection, preservation, and identification of the algal samples. So it's varying and it's specific to each of the member of the classes of algae. If suppose you are going for collecting chlorophycin members and preservation, it's different. And collecting anaphysin members, that's VGA, it's different. So collected from various collection sites, and it was filtered through plankton linear inch. So you have to make use of a phytoplankton net as you go for collecting the phytoplankton from weight. Then these phytoplankton and preserved in 4% formalin. And micro-transact method was described by, and it was also followed. And Edmondson system was also followed. And we also use this counting chamber to count the phytoplankton flora. And for identification of chlorophycin members, the water samples were collected during favorable period seasonally, but temperature should range between 15 degrees centigrade to, but if suppose they are collection of samples of anaphysin members, the temperature was referred for members of like Prescott, Philippus, Philus, and Habi. For identification of desmids, that is from among chlorophyci, I've got Cosmerium and Clostridium, which act pictures. Showing interest in make this practice of taking even camera Okay, next slide. Yes. So to identify the members of Sanophysis, samples will be collected from freshwater habitats at different seasons, and 3 to 5% formalin, and little of glycerin will also be added. And for studying the material under microscope, semi-permanent preparation in glycerin was made, and various blue-green species will be taxonomically determined with the help of standard literature like Giltler, Desikachari, Prescott, and Cormorant. So this is how few of our scholars have gone for collection, and the samples must be collected in the early 5:30 to 6:30, and the sample should be collected on the same day, same time from almost all the lakes of your study site. And this is because you know that the algal samples which are collected or the water samples which are collected early morning will give you good results, because the algae early morning will be towards the surface waters as you see for sunlight. And as the day is extending, reaching to afternoon, cannot hear. And as you know, the day extending to afternoon, and what happens is the algal members sink to the water. So that is the reason why and how we have to go for collecting the water samples, and such scholars have gone out for collecting the water samples. This is a field analysis. So as you go out for collecting water samples, you need to have a kit with you. You material like thermometer and Secchi disc and a device to scrape off the algal members if present on any substrata. And also you can have a forceps so that you can, take off the algal member if present on any of the softer substrata. You know that algal members are being found even on rocks attached to substrata. So this is how you should have an equipped material or kit with you. And as and when you collect the algal material, you need to take some notes. That is date of collection, time of collection, name of the lake, and site where from you have taken. And this is how one of our scholar, she's on site, she's taking for temperature using a glass thermometer. And color and order was carried out by sensible method. And pH meter was there where you can see for pH of the water body. And a sample for dissolved oxygen was fixed using this Ringler's reagent. And depth and transparency of each station would be measured making use of a Secchi disc. So Secchi disc is where it should be immersed into and take for transparency status of that particular lake. And algal diversity. So frequency of this diversity of algal species was done using this method, that is one drop method by Pearsall. It was proposed by Pearsall. And we have gone for evaluating the status of pollution lakes. And it was based on NIAGA's algal indices. So each of the lake is in which state? Is it in eutrophic state or oligotrophic state? So based on that, we have to give suggestions to the people living in and around, whether it can be used or it could be used for drinking purpose, or we have to count for the phytoplankton. This is a nearby chamber. It is having a counting area long, and the whole scale is divided into 9 big squares, and each square is 1 mm long and 1 mm wide. So this is how we have gone for following for 1-drop method sample onto a slide and put it onto this counting grid, and then you can see to the algal members within 100 liters of water collected per 1 gram. So out of the phytoplankton which we have assessed, where we have gone for abundance calculation, PHYSICO and PHYSICO. Chlorophysi was represented by 50 species, Bacillophysi was represented by 41 species, and Cyanophysi was represented by 29 species, and Euglenoophysi was represented by 10 species. So this is the record of phytoplankton we have taken from all different lakes of our study site. So seeing to our composition, chlorophysi members in different lakes. So here plus indicates presence and minus indicates absence. So we have got Actinastrum gracilium present in all different lakes, all 6 lakes, and Ankistrodesmus present in all 6 lakes, and Cara vulgaris also present in all 6 lakes. And we have got Chlamydomonas present in all 6 lakes, but absent in all 5 lakes, absent in Alisagar Lake. So like that, we have got Chlorella and Cladophora, Clostridium and Posmerium, and Coleochaete present almost in all the lakes. And Coleochaete is present in first lake and last 2 lakes, and absent in Aracheru Lake and Ashok Sagar Lake. So this is about the chlorophysian members, the data which we have collected and presented here. So along with this, in addition to chlorophysian members like Cara, Coleochaete, and so on, even Eudorina was present in all 6 lakes. Hydrodictyon was present in all 6 lakes, and Edogonium was present in all 6 lakes. And Pediastrum was present, and Cynidesmus was present, and Pandorina was also present. Volvox was observed. Ulothrix was present. Zygnema was present. So these are all the members of chlorophysian members which we have detected from 6 lakes of Telangana state. So Bacillophysi members, we have got Amphora from Eracheru Lake, Singagupalam Lake, Karakonda, Ramavaram Lake, Ashok Sagar Lake, and Alisagar Lake. And we have got Cycloptella present in all lakes. And we have got Fragillaria present in Singagupalam Lake, Karakonda, Ramavaram Lake. And we have seen to Gomphoneema present in only 1 lake, that is Alisagar Lake. And we have got Melocera granulata present in all lakes. And we have seen for abundance of Navicula in all lakes. So this is an account of Bacillophysi members, that is diatoms. And we have also seen for abundance of Pinularia and also Synedra and so on. And Euglenoophysi members, very few were observed. So we have seen Euglena present in Komacheru Lake, Singagupalam Lake, and Ashok Sagar Lake. And we have seen Phacus orbicularis present in all lakes. And we have seen for Trachelomonas present in all lakes, and so on. Members of Euglenoophysi were collected like this. And Cyanophysi members, we have seen for abundance of Anabina in 3 lakes. And we have seen abundance of Lingbaya in almost all lakes, Microcystis in first 3 lakes, and Oscillatoria which was present in 3 lakes like Ashok Sagar, Ali Sagar, and Karikonda Ramavaram Lake. And cytonema was present in first 3 lakes and absent in Karikonda Ramavaram Lake, Ashok Sagar, and Ali Sagar lakes. And spirulina giganteum was also present in all lakes, and Tolipothrix was also present in all lakes, and Trichodesmium erythraeum was also seen in few lakes. So these are the physico-chemical parameters which we have assessed, and all of these are expressed in milligrams per liters except for pH. Ordered in Karikonda Ramavaram Lake was highest, and turbidity recorded in Eracheruvu Lake was highest, and total suspended solids was highest in Eracheruvu Lake, and pH as it is in Singabupalam Lake was more, and dissolved oxygen was 9.6 in Komacheruvu Lake, and biological oxygen demand was 8.9 in Karikonda in Komacheruvu Lake, and COD was 4.5 in Ashok Sagar Lake, and chlorides estimated was highest in Singabupalam Lake, and phosphates estimated was highest in Komacheruvu Lake, and silicates estimated was highest in Singabupalam Lake. So this is how we have assessed for physical parameters. Next. Next. So the same thing is shown in graphs: transparency, turbidity, temperature, total solids, and total dissolved solids, and pH, which was expressed in milligrams per liter. And Karikonda Ramavaram Lake, it is 87 transparency, and turbidity in Eracheruvu Lake is 26, and temperature recorded was highest in Komacheruvu Lake, and TDS was highest in Karikonda Ramavaram Lake, and pH is also highest in Singabupalam Lake, that is 8.5. So this is the graphical representation of all the physical parameters we have assessed. So dissolved oxygen comes to around 9.6 milligrams per liter in Komacheruvu Lake, and BOD is around 8.9, and it is in Komacheruvu Lake, and COD was highest in Ashok Sagar Lake, 4.5, and estimated chlorides was more in Singabupalam Lake, that is 103.5, and phosphates estimated was more in Komacheruvu Lake, and silicates estimated was more in Karikonda Ramavaram Lake. So this is a graphical representation. So phytoplankton community, which was abundant, is also taken for statistical analysis. Like correlation coefficient was employed for correlating the relative significance of various physical and chemical parameters. Okay, next you can go. Yeah, water quality index. So water quality index, previous slide. Water quality index is a significant tool, and various methods are available for assessment of water quality index, where we can go for comparison of physico-chemical and biological parameters as reported by Horton, Landwehr, Dunning, and Steenhartel, Heod, and Canadian Council of Ministers of Environment. And the final water quality index value was determined based on annual average value of every year taken from each site. So this is a formula where you can go for assessment of water quality. And as a resultant, we have got the result. So based on the quality index, if it ranges from 91 to 100%, we say that water quality is excellent. And if it's ranging between 71 to 90%, you say that it's good. So based on this, we have got results saying that water quality of Komacheruvu Lake was medium, as it shows 66%, and Eracheruvu Lake is good, as it shows 72%, and Singapore Lake water quality was medium, as it is quality is good, and Ashok Sagar Lake water 60%, and this indicate that as water quality decreases, the pollution level increased. So every year, as you go for assessing the quality of water, you can understand for the pollution status of each lake. And this is maybe due to population growth, that is population explosion, and also environmental discharge, that is from domestic sewage, and also surface runoff of crop fields. So this is how you can assess for quality of water in each of the water body. So graphical representation of phytoplankton, which we have shown before. So we have got 38% of chlorophycin members, and followed by 32% of bastrophycin members, followed by 22% of salophycin members, and 8% of euglenophycin. So chlorophycin members are more abundant, and compared to salophycin members in the lakes we have assessed. So there are some indices to calculate for diversity. And we have got Margaret Diversity Index, Shannon Diversity Index, Simpson Index. So Margaret Index shows differences so that the number of individuals is less important for calculation. And based on this, Margaret Index was ranging between 12.20 to 12.87, and the lowest value was for Ashok Sagar Lake, and highest value was for Eracheru Lake. And according to Shannon Diversity Index, it's commonly applied to biological system for calculating diversity, and it's obtained for mathematical analysis by Shannon. And the value of this index ranged between 1.2 to 1.21, and the lowest value was at Komacheru Lake, and highest value was at Alisagar Lake. And Simpson's Diversity Index is a measure of ecological diversity, and it's used to quantify the account of diversity, and it was introduced by Edward Simpson. And the value of the Simpson Diversity Index ranged between 0.33 to 0.84, and lowest value was found at Karakonda Ramavaram Lake, and highest value was recorded at Ashok Sagar Lake. So to assess the trophic level of each lake, we follow for NIAGAD's indices. And NIAGAD have proposed for 5 indices of biological monitoring. And it's based on the individual population of Sanophysi, Chlorophysi, Bastrophysi, Euglenophysi, and combination of all population together. And based on this, you can also calculate the status of eutrophication. And lake is classified based on their richness of nutrients. So you can say that it is oligotrophic, and here poor and have concentration. And mesotrophic lakes have good clarity and average level of nutrients. And eutrophic lakes are nutrient deficient, and ultimately unproductive, and detritus-free systems, and developed ones with increasing amount of nutrients, and thus leading to organic materials. So several authors have followed this trophic status of lakes based on the formula of NIAGAD's indices. Yes. So here what we have observed is saying to this formula of NIAGAD indices. In Karakonda Ramavaram Lake, it is 9.09, where you say that status is weak trophic. That means reaching to trophic state. And Komacheru Lake, it is 5.27, and you can declare it as oligotrophic. And Eracheru Lake, it's 5.41, it's oligotrophic. And Singapore Lake, it is 6.64, it's weak eutrophic. And Ashok Sagar Lake, it is 8.53, weak eutrophic. And Alisagar Lake, it's 10.86. So as it's eutrophic in nature. So we have got one index, Palmer's algal index. So for finding the water sample as high or low organically polluted, the algal generic pollution index of Palmer was followed. And here in the present study, Palmer's algal genus index was used to study the water quality of all 6 lakes. And a list of most pollution tolerant genera which belong to 4 groups of algae according to Palmer's index was calculated. And total value of algal genus pollution index of 6 lakes were 30, 45, 35, 40, 28. And is stated that it's highly or high organic pollution. It's confirmed that the values indicate that it's highly organically polluted. So this is a correlation matrix of physico-chemical and phytoplankton, and we have to correlate all these factors to all the different classes of algae like Chlorophyceae, Basilorophyceae, and you can say whether it is significantly related or it is positive correlation or negative correlation. The Chlorophyceae members were significantly correlated with physical factors like temperature and TDS, pH, dissolved oxygen, biological oxygen demand, phosphate, and negatively correlated with transparency, COD, chlorides, and silicates. So thus you say that C members are significantly positive correlation, that Chlorophyceae significantly for positive correlation with members of Basilorophyceae and Sanophyceae and with euglenoids. And Basilorophyceae were significantly pH, alkalinity, ammonia, phosphate, free CO2, and primary productivity, and negatively correlated with transparency, TSS, DO, COD, and silicate. So thus, Basilorophyceae members show significant positive correlation with Sanophyceae and negatively with Euglenoophyceae. And Euglenoophyceae show significant positive correlation with transparency, pH, COD, chloride, silicate, while negatively correlated with temperature and total solids. And Euglenoophyceae show negative correlation with Chlorophyceae, Basilorophyceae, and Sanophyceae. Sanophyceae are positively related with temperature, TDS, TSS, pH, and DO, whereas negatively correlated with transparency, COD, chlorides, phosphates, and silicates. Thus, Sanophyceae members have shown significant positive correlation with Chlorophyceae and Basilorophyceae, and were negatively correlated with Euglenoophyceae. So this is according to our correlation matrix. And abundant growth of these phytoplankton was in comparison to all the parameters assessed. So as I'm reaching time, here I would like to show for some of our microscopic observations. So from Chlorophyceae, we have collected Ankystrodesmus, Cara vulgaris, Chlorella, Cladophora. And as you know, Cara is second reproductive, and Chlorella as SCP, that is single cell protein. It's also used in space travel. And Cladophora is a member of Chlorophyceae, where it is showing for branched and filamentous thallus. Next. So we have also collected for a few other species of Clostridium. So Clostridium is one among the desmid. Clostridium is usually called as an unconstricted desmid. Here it's unicellular, and whole of the single cell is divided or divisible into 2 halves. Each half is called, or each half is called as a semicell, and it has got chloroplast. So this is about the morphology of Clostridium. Next. So few other members of Chlorophyceae like Coleochaete and Cosmerium was also collected. So as Clostridium is unconstricted desmid, you say that Cosmerium is constricted desmid. Both of these serve as best examples for desmids. So we have also collected Eudorina, Eudorinia is colonial, where we say that thallus is cenobium, and hydrodictyon, where we say popularly called as waternet, and Edogonium, where we have got two different forms like Macrandrus and Anandrus. And we have also collected Pediastrum, Cinedesmus, and Spirogyra. And Pediastrum, you see for the arrangement of cells, we have got a centrally located cell surrounded by two concentric rings of 5 cells each, and peripheral cells which are more in number. And cells of the peripheral side are different from cells of the central cell, where they show for some processes which are called as bristles, and it's non-motile. And Cinedesmus is also non-motile alga, and Cinedesmus can be employed in application of phycoremediation. And one of our scholar, Dr. Ramesh, has employed this Cinedesmus into application of wastewater treatment, where he has taken sugar mill effluent. And we have collected diatoms like Cycloctella, Fragilaria, Gomphanema, and also Navicula, Nischia, and Pinularia, and Staurensis, and so on. You have Spulina and Lepicin. Collect Anapinas and have and also seen gliotrichia and microcystis and nostoc. And also collected Oscillatoria and Crucocus. Lingbia and also Tolipothrix was collected. Microscopic observations, we understand that we could collect PHYSI in more, and members of PHYSI to an extent, and members of PHYSI BGA will act as a tool for measuring the assessment, measuring the quality of water. They also act as bioindicators or algal indicators. So based on this, let me conclude saying what is biological monitoring. Biological monitoring is a water quality management tool, and the process of biological monitoring is important because community structure can be altered with any change in their environment by the effect of pollution, which could be easily missed by conventional and physical indicators. And biological monitoring aims to monitor the ambiency of water quality, and each and every lake in our country is associated with many problems which are needed to be addressed, and water quality plays an important role, or it's a factor, and the primary cause of deterioration of surface water quality is due to some disastrous effects, and it is mainly due to disturbance caused by municipal domestic wastewater and industrial and agricultural and solid and solid refuge. So one need to understand this mechanism or a tool of biological monitoring a water body clean. And in this biological biomonitoring, planktonic algae will play an important role, and are used for biomonitoring because of their nature of cell structure. So the intracell is different, and are totaling almost all concentrated metal ions from ambient waters. So thus exists a relationship between metal absorption and its toxicity to microalgae, which is become more important. And it's believed that apart from the scientific investigation, periodical monitoring and details of micro and macrobiological organisms, the socioeconomic conditions of people, population growth, agriculture and industrialization in the city, urbanization and land use pattern are also significant for improvement of certain principles and guidelines for safety of So based on this principle of biomonitoring, saying that the lakes which we have studied were eutrophic in state, that is, they were enriched with nutrients and found suitable for luxuriant growth of the algae. To conserve biodiversity of phytoplankton, such state of lake will play a significant role. And the lakes studied were of scientific investigations, and suggestive measures are to be taken where you need to control pollution and to have a check over dump of sewage and also dump of industrial effluents from surrounding area. That is, people living in and around should be educated not to dump the waste or the sewage into the lakes, where you should not see that, or you should see that it's not polluted. And regular monitoring of these should be under the control of Telangana Pollution Control Board. And we are thankful to our government of Telangana, who have shown interest in restoration of lakes. And this is all giving assurance to provide water, majorly for irrigation, drinking, and other purpose to people in our state. So thus they have started a program which is called as Mission Kakatiya. And as I have completed my talk, maybe it was interrupted, and we have come to an end. So based on our work, last year, very recently, we have presented this work at 107th Indian Science Congress, and his scholar, Dr. Ramesh, where both of us have done a poster and we have presented. And the title was Studies on Phytoplankton Assemblage and Macrophytes, different water bodies in and around Nizamabad district. So I just wanted to show this as it is a recent work. And finally, before concluding, I should express my due thanks or acknowledge due thanks to my guide and mentor, Professor Vidyavati Madam, who is former Vice Chancellor of Kakatiya University. And she's inspiring. She's a source of inspiration for all the research scholars, all the scientists. And she's called as Mother of Kakatiya University. And I'm very proud to be a scholar of Madam Professor Vidyavati Madam. And she has crossed age of 80, but still she's active, energetic, and hope that God will give her more energy and stay with us and support us continuously. And coming to our department, we have got PhD awarded to 5 scholars. So Dr. Ramesh is awarded, and now he's serving as NEET faculty at Karnataka, Davanagere. And Dr. Shailaja has completed her PhD, and she's serving as faculty at Government College, Siddipet. And Dr. Samira has completed her PhD awarded, and she's serving as a faculty at Government College, Golconda, Hyderabad. And Dr. Srinivas is awarded. He's serving as a faculty at Government Degree College. And Dr. Rajalakshmi has awarded, and she's serving as faculty at Kottagadam Singareni Women's College. Achievement from our department. And a few more scholars are pursuing under my guidance. He's Mr. Arjun, and she's Mrs. Bhargavi, and he's Mr. Raju and Mrs. Sweta. So this is the ongoing work in our department. And thank you all for patient listening. Thank you.
Dear participants, please, if you have any queries, put in chat box.
That cells are
Thank you for wonderful presentation, ma'am. Thank you. It was very colorful and so many new concepts we have come to know about the evaluation of various parameters and how the lakes can be classified into various forms. It was wonderful to see so many concepts. What to be done in hydrobiology and aquatic ecosystem study. Thank you so much for such a wonderful and beautiful presentation, ma'am. Thank you so much. Now, Yadav sir, can you start sharing your screen?
I will share my screen. Is it okay? Hello, sir.
Yes, it is visible, sir.
Visible. How much time you have given me?
Till 12:30.
12:30. Okay. This lecture is about 3 hours lecture, but I will be winding up in this 1.5 hour. I mean, not 1.5 hour, but yeah, 1.5 hour. And I will run through slides. Really, those who want to study grasses, they need to spend 1 or 2 days with me. And my laboratory is always open. Those who are interested in knowing grasses. Now let us go for this group, very interesting group, amazing group of flowering plants on this planet. Grasses, an important underutilized natural resource for sustainable bioeconomy. This is the title of my lecture. I will be introducing various types, various genera, not at species level I'll go. I will just introduce you how to identify genera and why grasses are so important. You know, grasses are considered to be centers of civilization. You know, human beings, we were like chimpanzees in forest areas, but afterwards we have moved to open lands in grasslands, and where we established ourselves, and these grasslands became a major, they have played a major role in our civilization. And you know, most of the civilizations, world's well-known civilizations were developed in a grassland areas. So these are centers of civilization. Another thing which I want to tell you, I call it as a green gold of earth. I think the black gold is our soil, green gold is our grasses. And grasses, a great group of plants of this planet. Unfortunately, all of us, we take them for granted, and I consider grasses as a saints. Rishi Munis, they never try to look beautiful, but they serve the entire world. And our 80% of requirements of human beings, remember, 80% of human requirements come from grasses. Your sugar, your food grain, your fodder, your milk, your meat, all that are grass products. And therefore, grasses, although they grow everywhere, they are in abundance, but usually we don't respect them. And these grasses play a major role in our life. Now see, for this shepherd man, what is a green gold? Grasses is a green gold, and his entire economy depends on grasses. Then, you know, in Kolhapur, we have Varna ice cream, we have Gokul ice cream, we have Gokul Good Dairy. Because around Kolhapur, we have a lot of grasslands, and because of it is one of the most important world famous place for production of milk and milk products. Similarly, Kolhapuri chappal is also a grass product. Similarly, Tambra Rasa, Pandra Rasa, which is known in Kolhapur, is because we have goats which are healthy, and we have beautiful mutton in Kolhapur. So what I want to tell you, our civilization, our thinking, our every walk of life is governed by grasses. And now all of you should know that grasses only little you try to remember. There are out of 12 subfamilies, 10 subfamilies are reported in India. There are 266 genera of grasses in India out of 711, and there are 1500 plus grasses out of 12,000. So grasses, 1/3 of grasses of the world are found in India, and that way we are really one of the richest country in grass diversity. Now you see, just I will go run through slides. Grasses and food. I need not to explain you these things. Grasses and fodder. Grasses and cover of mother of earth. You know, because of grass cover, the soil is kept at a place and we conserve soil. So that way, grasses play important role as a cover. Grasses and soil conservation. Grasses and restoration. Okay, if you want to restore any ecosystem, first you have to go for plantation of grasses, growth of grasses, then only we can restore any degraded ecosystem. Grasses and wildlife. If you want to save tiger, you have to save our grasslands and grasses of the region, because herbivores live on grasses and tiger lives on herbivores. So it is a grasses you have to conserve, then only you can conserve our charismatic animals in the wild. Grasses and bioremediation. You know, many grasses now they are used for bioremediation, phytoremediation, what we call. So that way also they have great role. Grasses and climate. Okay, so they produce maximum biomass and absorb carbon from atmosphere. Grasses and sugar. You know, our Kolhapur region is known as sugarcane lobby, and the entire politics of Maharashtra is governed by single species, that is Saccharum officinarum. Grasses and medicine. Some grasses definitely are of medicinal value. Grasses and aromatic oils. You know, symbopogons, they have aromatic oils. Grasses and biodiversity conservation. You know, if you want to conserve biodiversity, we need to grasslands, which are breeding places for birds, lizards, snakes, all insects. And that way, if we want to conserve biodiversity, grasslands are of great importance. Grasses and biomass production, grasses and shelter, grasses and paper industry. Our bamboos are used for paper industry. Grasses and alcohol industry. Now we have started making alcohol from grasses or sugarcane. You know, grasses and microbes. Many microbes are associated with grass root, rhizosphere, with many microbes which play important role in maintaining health of the soil. Grasses and ornamental plants. Some grasses are of ornamental plant. I told you, grasses don't try to look beautiful, but they serve you in all sense. Grasses and landscaping, grasses and civilizations, grasses and bioenzymes. You know, harmful grasses, there are harmful grasses, there are poisonous grasses, and grasses, there are wild relatives of grasses which are of great importance in food production. Grasses and religion. Our many grasses like our Cynodon dactylon, and many are there which we use in a religion. So they are spiritual grasses. Grasses are as a bioindicators. Many grasses show health of the soil. Grasses and dairy industry. Grasses and meat production. Grasses and ecosystem health. Probably must have understood with these 2-3 slides that grasses is a major bioresource of this planet, and we must know grasses. I know all teachers in colleges, students run away from grass identification because the spikelets are very small and identification goes difficult. But I will make it easy. It is not that difficult as you feel. We can identify grasses. Now grasses may be annual or perennial. You can see this root system. Grasses have inflorescence, then leaf, then sheath, then you have node, then we have internode, then we have fibrous roots. Okay, and the terminology, which is very important, very different terminology, we use the sheath, the lamina and sheath. which covers encircle stem. This is the seed. And in between leaf, lamina, and sheath, there is an outgrowth known as ligule, which is a peculiar character of grasses not found in cypresses. And this is the stem. So see this auricled auricle. So different kinds of ligules you can see, hairy ligules. Then this is a serrate ligule, papery ligule, and only few grasses are there where there is no ligule, and that is the most primitive grass. So you can identify grasses based on types of inflorescences. The types of inflorescences are of great significance in identification of grasses. So panicle, raceme, spike. So these are the types: true panicle and false panicle. This is false panicle, this is true panicle, solitary spikelet, and these are the group of spikelets. So this terminology in 1 hour I cannot teach you. We need practical class, then only I can teach. So this is a false panicle, right? False panicle. Then raceme, you can see this raceme, pedicellid spikelet sit directly on the rachis. These are racemes. Then spikes, you can see this is a spike. Sessile spikelet sit directly on the rachis. This is a rachis, and these are sessile spikelets. Grasses flower. Flower structure, of course, stamens and carpels provide no diagnostic character. At the base, we have laudicles. Then we have laudicles are considered to be perianth, ovary, stigma, and stamens. Usually 3 stamens, 2 styles, bicarpellary ovary. And we have this rachilla. And then after rachilla, we have lemma, we have pallia, and then we have flower. Okay, lemma, pallia. The lowest bloom, glooms, they are known as glooms, and these are lower gloom and upper gloom. That is the terminology. And then all are lemmas. So try to understand this. I will send you this presentation to you, and you can learn. Okay. So see, in morphology of spikelet, lower subtending bloom, upper subtending bloom, lemma, pallia, laudicles, and stamens. Okay. Then another flower, a lemma, pallia, laudicles, stamens, and carpel. Okay, gynosia. So this is the way it looks, and that is panicoidy. We identify this as a panicoidy, where there are spikelets with more than 2 flowers. Here, spikelets are with single flower. This is single flower spikelets, and these spikelets fall down. I like this, this green colored. So, and leave lower glooms on the rachis. That is a pooidy. And here, the entire unit, along with lower glooms, fall down. That is panicoidy. So Bohr has identified 2 subfamilies, panicoidy and pooidy. Of course, now there are 12 subfamilies, but Bohr's classification I find very practical for identification of grasses. Okay, so systematic treatment: poaceae, panicoidy, and pooidy. Spikelet 2 flower, falling entire at the maturity. Pooidy, spikelets many flowered, spikelets falling living blooms. Right. So we will not go in much details, but panicoidy is again divided into 3 tribes: Andropogony, which has awns, Panicae, and Medi has unisexual flowers. Okay, unisexual flowers. So these are the genera which are found in Maharashtra, and pooidy is directly divided into tribes: Aliroidy, Agrostidae, Aristidae, Arundinidae, and so on. 25 this one. Now, see, this identification of grasses is not that difficult as we think. If you start developing one practice, that every time when you see grass spikelet, see with the lens, hand lens, which will cost you only about 100 rupees, around 100 rupees. With that lens, you can identify all the grasses if you have spikelets. Now see, this is a, so you see the boat-shaped bracts. This is Apluda. This is the endopoa. You can see this endopoa with the lower glooms, and then these are the spikelets, and then they are on. Then you can see Isacne. All Isacne will have spikelets like this. Even if you see shape, size, and arrangement, immediately you can identify Isacne. Then Syrtococcum. See the spikelet, shape of the spikelet. It is oblique. One side it is extended, so it is a Syrtococcum. This is a Thymida. This is Pseudanthristia. You see the lower gloom, Glypocloa. Just you see lower gloom, you can identify Glypocloa. Coix, you can see this is a female flower. These are male flowers. You can identify all members of Medi. And this is a Glypocloa again. You can see lower gloom and tragus. Now these photographs itself indicate that it is not difficult. Only they are small in size, and we neglect them because of small size. So you can see this Chionacne, Apocopsis, Arundo, Arthraxon, Avena, Segencrus, Centotheca, then your Arthraxon, then your Arthraxon and Acracne, Coix, Acroceros, Chloris, Alloteropsis, Symbopogon, Dicantomis, Syrtococcum, Stiphagrostis, where the on is, plumose, and then we have Symbopogon, we have Chloris. Now see these photographs, which I have taken with a simple camera also, and today you have beautiful mobiles. Why not to develop a hobby of photographing grass spikelets with your mobile? I think, and you can identify all. Will you get confused with all these? I don't think you will get confused. They are so unique, and we can identify just by looking at spikelets. Okay. Similarly, Arthraxon, Theliopogon, Manissuris, Glypocloa, Hachelocloa, Tragus, Ichimum, Ilionurus, Bathyriocloa, Lircia, Lopopogon, Sencrus, Rinchelitrum, Tetrapogon, Glypocloa, then Glypocloa again. You can see this lower gloom, very beautiful lower gloom. So it is not difficult. Okay, grasses is the most important group as far as economic importance is concerned. Similarly, biodiversity conservation, phytoremediation, in all these, our grasses are going to play a major role. And therefore, dear biologists, all of you should try to know grasses of your region, and they are playing important role in many aspects. So you can see this Ilionurus, Glypocloa, Manissuris, Opuras, Dicanthium, Theliopogon, Tragus, Centotheca, then your Lopopogon and Vitiviria. You tell me, just I am showing you lower gloom of these spikelets, and they are such a beautifully ornamented and beautifully designed that you can identify them without any difficulty. Right now, so just I introduced, grasses are not that difficult. Don't get annoyed with grasses. Don't run away from grasses. Try to go near, and I am sure one day you will be knowing majority grasses of your region. Now I come to the latest classification of family Gramineae or Poaceae. Now you see the Gramineae starts with sister group is this one, Anomocloidae. This is the Anomocloidae. From there our grasses start, and Anomocloa is found in America. So from here we start. Then another family is Pharoidae. We have only one genus of Pharoidae in India. Then Pluleoloidae, then Orhizoidae, Bambusoidae, Poidae, Aristoidae, Panicoidae, arundinoide, micraroide, danthenoide, and chlorodoide. Now you see, evolution is this way. This way, grasses have evolved on this planet. Lot of molecular work has gone in, and this is the, you can say phylogram and evolution of grasses. The most advanced subfamily is chloridoide, and most primitive is anomochloroide. Just for your information, I am showing you because you should know these diagrams explain you evolution of grasses and C3, C4 mechanisms which have evolved, and you see all these are C4 grasses here. You see all these are C4 grasses, and these are the primitive grasses. So this diagram tells you a lot. Okay, so anomochloroide, paroide, pulioide, oryzoide, bombusoide, pooide. This is a BOP clade. Aristoide, then our panicoide, arundinoide, micraroide, danthenoide, chloridoide, PAC mad clade. Okay, so it is a after these words, P means panicoide, A means aristoide, then C may A. So depending upon these letters, they have been said as a PAC-MAD clade and BOP clade, bombusoide, oryzoide, and paroide. That is this clade. So these are the clades which we recognize. Now this is a evolution. POAC, anomochloroide, I have told, and the latest is this PAC-MAD, panicoide, aristoide, chloroide, micraroide, arundinoide, and danthenoide. That is PAC-MAD clade and BOP clade. So this little information about classification. Now, this is that grass which is the most primitive and sister to all the grasses of the world, and its name is Anomochlora maranthoidea. So it is a most primitive. Just I showed it does not occur in India, but I thought that you should know it, so I have given. And paroide, we have Leptopsis and Paris, which is found in India. So these are two subfamilies, but we don't have Leptopsis. Then Pulioide, this is a Pulioide, and this is the genus which is found in, it is African genus. Okay, so these are most primitive subfamilies with few genera. Anomochloroide has 2 genera. Then paroide has 3 genera, and Pulioide have some 4-5 genera. So this is the about 3 genera. Pulioide has 3 genera. Okay, so this now let us go to Oryzoide. After these 3 subfamilies, 4th subfamily comes, that is Oryzoide, in which we have Streptogyna, Ehrarchta, Lyrcia, Oryza, Hycorrhiza, and Zizania. I will show you one by one. Not all photographs I have, whatever I have studied, that I will show you. Now see, this is a Oryzae, and this is a genus Lyrcia. Marshy places, you get the genus, it is a wild relative of Oryza, genus Oryza, and many genes we can use for improvement of our rice. Okay, all characters I will not explain, only I will go through important characters. Now, see, there are about 20 species in this, and it is a very peculiar. Now you see, just like rice, it looks, and on margins you get bulbous based hairs, just like rice. And here, the lower glooms are reduced, almost reduced, they are not apparent. So whatever you are seeing, these two, these are lemmas and pallias, right? Spikelate, lemma and pallia. There are no lower glooms in this group. Okay, Oryzae. Now, Oryza is known to you, rice, which is a staple food for 1/3 population of the world. And this grass has played a major role in our life. Now you see, this is lower gloom, this is upper gloom, and these are lemma, this is a pallia. And we have so much varieties of rice I need not to explain you. So this is the way we can identify rice. Okay, rice. I think everything is clear to you with my slides. Okay. Now, another Oryza rupipogon, which is wild relative of rice. Oryza rupipogon. Oryza rupipogon. Which is a wilder relative, I want to tell you. Then Portracia, it is a tetraploid. It is initially it was included under Oryza, but now it is separated into Portracia, Portracia, Portracia cortacta. This grows in a brackish water in muddy soil along the seashore, and it has great tolerance for salinity. So this can be used in the improvement of our grass, that is Oryza sativa. Then another is Hygrorhiza. This is a Hygrorhiza. See the leaves. They are spongy. They grow in the water. All Oryzae are water-loving plants, and you can see this spongy petiole and leaf blade. So this is monotypic genus having only one species, that is Hygrorhiza aristata. Okay, little efforts if you make, you can identify. Now Bambusi is a big group. There are more than 25 genera and about 125 species in India. This is Bambusa vulgaris and this is Bambusa arundinaceae. Okay, it is little bit difficult group because we don't get flowering. So Dendrocalamus, this is Dendrocalamus strictus, where these are arborescent grasses. And this is, you can identify this bamboo very easily. You see the seed. Seed is of Pseudium gava size, about 5 centimeter in diameter. Okay, so this is a Molucena bacifera. Molucana bacifera. So this is a one grass which you can identify, a bamboo which you can identify without any difficulty. Melocanna bacifera. Right, now we come to sixth family. Bambusoideae I have told you. Now sixth family is Phoidae, and in which there are many tribes. That, for example, Tritici. So we have Triticum feet, which is known to you, and remaining 3 genera I will show you. This is Aegilops, which is one of the member which has provided genes to improvement of our feet. Aegilops. Okay, this is Aegilops. See the spikelets, and that can explain you a lot of things, and you can identify very easily. Then, right. So solitary, laterally compressed spikelets, you can see, right? Now, see this Triticum. Triticum estiva, which is well known to all of you. Feet is known to all of you, and feet is another grass which feeds our 1/3 population of the world. Okay, human population. So this is the wheat. You can see this grain also. There are a lot of characters to identify, but in in short, I will be explaining you this Triticum. There are 10 to 20 species of Triticum, right? Now, another tribe from subfamily Phoidae is Poi, Avena. This is Avena sativa. Okay, Avena sativa. You see the lower glooms, see the spikelets, and see the awn. Okay, nobody can go wrong in identification, right? Avena. Then this is Helictrotrichum skimdi. Helictrotrichum skimdi. This is a, see the lower gloom, upper gloom, then lemma and palea, and see lemma has on backside. So this is a Helictrotrichum. Then this is Phalaris, which is a weed of wheat crop. And in young stage, it looks exactly like wheat. You can see this wheat and this is Phalaris. Phalaris, which is common in wheat fields, especially in Delhi side, and that way it is a major weed of crop plants, right? Then you can see Lolium. This is a Lolium temulentum. Lolium temulentum. On one side, I have given characteristics, number of species found, distribution of the genus that you read. I will send this presentation to our Dr. Hamid, and he will send it to you. Okay. Then monotypic grass genus Dactylis is a Dactylis glomerata, which is a monotypic grass. Right? Monotypic grass. Nobody can go wrong in identification. This is Poya. Only in temperate region we get Poya. Nilgiris. In our area, in Nilgiri area, we get 2-3 species of Poya. There are about 15 species in India, especially in Himalaya, and it is cool temperate regions are preferred by this genus. Now this is Poya vivipara. You know, this is an interesting grass. Of course, it is not from, it is from Himalayan region. You see all spikelets get converted into bulbils, all bulbils, and it only propagates through bulbils, and therefore it is known as Poya vivipara, right? This is Sclerocloa. Now you see on one side 291 number is given. It is according to Kellogg's classification. C has given number to the genera. It is 291. There are 711 genera. This is a 291 number, Sclerocloa. Of course, it grows in a Kashmir region. You can see this Sclerocloa only by looking at habit also. I think you will be able to identify Sclerocloa dura. Okay. Now let us come to 7th family, subfamily Aristidoide. Very unique family, and all of you will be able to identify Aristidoide, Aristida, Spartidia, and Stiphagrostis. These are the 3 genera found in this subfamily. So what are those? Now you see, this is Aristida, and the lemma on often fused to form a twister column, and it is a triped. You can see this triped, lower gloom, upper gloom, and this is a lemma which is triped at the apex, triped at the apex, and that way nobody can go wrong in identification of genus Aristida. You see the spikelet, and if lemma is triped, you say it is a Aristida. Another, bundles, you know, there are vascular bundles. Usually in all grasses, there is only one sheath bundle, but here in Aristida, there are 2 bundle sheaths around the vascular bundle. So that way also it is. This is a Aristida funiculata in all grass, you know, Malran. We say we get this grass, and you see this lower gloom, upper gloom, lemma, which is triped at the apex. So this is a Aristida. Now, this Stiphagrostis is the same, more or less same, lower gloom, upper gloom, lemma, which is triped, but one limb is with plumose hairs. So this is Stiphagrostis, okay, which is grows in a Rajasthan region. Stiphagrostis. Now you can see very clearly lower gloom, upper gloom, and this the third on with plumose hairs. So you cannot go wrong in identification. Okay, so I have shown 2 genera from Aristoide. Now Panicoide is another subfamily. Now we have come to 8th subfamily, Panicoide. Chandrasekhariana, Jansinella, Centotheca, Arundinella, Garnosia. These are the genera in this family. This is Chandrasekhariana. It is a monotypic, and it is named after, this name is commemorative. Chandrasekhariana is a Chandrasekhar, named after him by Nair, Vijay Nair, and there is only one species, Chandrasekhariana keralensis. It is only property of Kerala. It is found in Kerala and it is a unique grass one should see. And see the lower glume, upper glume, lemma with the 2 tooths and one arm. So you can identify. So this is Jansinella. This was supposed to be monotypic grass, but we have described a new grass, Jansinella neglecta. So this is Jansinella griffithiana. Right from Burma to Western Ghat, in all mountainous places it grows, and it is a common grass in Western Ghats. You can see this lemma with the 2 awns, small awns, lateral awns, and the central awn is a dark brown, flat, and twisted. So you can identify it. This is one unique genus. Nobody can go wrong. Okay. So this is Jansinella griffithiana, which we have described. And then we have described one new species, Jansinella neglecta. You can see this. This is Jansinella griffithiana. You see the lower glume, upper glume, lower glume, upper glume, lemma, palea. Okay. And the seed here, this is the seed. So these are very distinct and we have described as a new species. Then this is your Lophatherum. It is a forest dwelling grass, Lophatherum gracile. And you see, this is a spikelet, and from the tip of spikelet, you get many hooked hairs. These are many hooked hairs by which it get attached to the animal fur, and that way the spikelets are dispersed. So it is, again, nobody can make mistake in Lophatherum gracile identification, right? So this is the Lophatherum gracile. From the tip, you get these hairs, and you can see these hairs, Lophatherum gracile, which are hooked, shaped, and with barbed hairs by which it get attached. So this is. Now this is our boom grass. We call it as a boom grass, Thyssalodina, which is also a monotypic grass. In grasses, there are many genera which are monotypic, and it is a very prominent grass in Northeast India. Spikelets are very small, and see on blooms, we have beautiful hairs, right? Just like our peacock. The hairs are there, and these are styles, and you can identify, see the inflorescence, panicle, it is a panicle, and you can identify it is monotypic grass. Now see, who will grow? Who will make mistake in identification of this grass? This is Centotheca lapacea, right? Which is again forest dwelling grass, and they have mechanisms for animal dispersal. See this lower glume, upper glume, see these lemmas, and these lemmas have bulbous based hairs pointed downwards. They get hooked to the animal fur, to the cloth of human beings, and they are dispersed. This is Centotheca lapacea. You can identify. Now this is Arundinella. Arundinella is a unique genus. You can see this lower glume, upper glume, and the shape of this spikelet. The shape of spikelet is very useful in identification of genus Arundinella. This is Arundinella leptocloa. Now see this different species of Arundinella. Arundinella nepalensis, Arundinella medci, Arundinella leptocloa, Arundinella ciliata, Arundinella, this species we have not. Purpurea, pumila, spicata, setosa, tuberculata. Now all these you can easily identify by just the shape of spikelet. How it looks, keep it in mind, and you will be able to identify Arundinella. Then this is a Garnosia, which is a forest grass. It grows on tree trunks, it grows on walls, on rocks, and so on. And it has a lemma is always on. So this way it is unique. See this Apluda. They have boat-shaped these bracts, and in that you have these spikelets, male, female spikelet, male spikelet, and very easily you can see, you can identify this grass. Again, this is a monotypic genus, has only one species, Apluda mutica. If you know one species, it is a common in all the parts of the country, and you can easily identify by these photographs. Then Arthrazon, another genus is Arthrazon, which is mainly probably originated in India, and it is diversified in India. Maximum species are found in India. Out of 27 species, 25 species are found in India. You can understand. So it is a Indian origin grass. You have a lot of scope for biosystematic studies. So you see this lower bloom. This is the lower bloom of different Arthraxon lanceolatus, variety echinatus, Arthraxon lanceolatus, variety lanceolatus, Arthraxon lanceolatus, variety ricei, variety villosus, okay? And all these varieties you can identify, and that way, that is Arthraxon. Chionacne, you know, Chionacne is a Medi. Medi means our maize group. Medi. Chionacne has beautiful structures. These are large, robust grasses. Leaves are just like maize. You can see these leaves are large, and you can see the inflorescence in the bract, axilla bract. Hello, Nampi sir. I am in lecture. I will phone you. I will phone you after half an hour. So this is the grass you see. Who will go wrong? Tell me. This is a female flower. These are the male spikelets. This is a female spikelet. And you know, this female spikelet, it has a hard coat, and inside there is a female spikelet. So this is a very unique grass. Nobody can go. Chionacne, Ponizi. And this is a Polytoca, Chionacne, another species of Chionacne. You can see this Chionacne, right? Chionacne, which has a different kind of this one. These are male flowers which have come out, and in the axils we have these female flowers. So you can identify Chionacne, right? This is a Chionacne. You see this. These are male spikelet, these are female spikelet, and you can identify different species. Then another is Trilobacne. So this is a Trilobacne, or now Kellogg has merged into Chionacne. This was considered to be monotypic genus. You see this. This is okay, male spikelets, and below male spikelet in the axils we have female spikelets, and this female spikelet has, you know, this lower bloom of the female spikelet is hard, shining, surrounded the rachis, three-lobed. This is a lower bloom, which is tri-lobed at the apex, and that's why named Trilobacne. And you can very easily identify this is only found in India. Okay, it is indigenous to India, endemic to India. Then another genus, Chrysopogon. None of you can make mistake in identification of genus Chrysopogon, because you see verticillaster type of inflorescence, then this capillary pedicels, and then there is a bunch of spikelets. Each bunch of spikelet has 3 spikelets. One in the center is a female spikelet, which is sessile, and lateral 2 are male spikelets. Like that, all Chrysopogon species have spikelet in 3s. 1 female, 2 male. Okay, so this is a Chrysopogon fulvus. So you can see this Chrysopogon velutinus, 2 lateral spikelet, central female spikelet. You see this callus. Then this is another aciculatus, female spikelet, 2 male spikelet. Then castaneus, you can see. Then your grillus, then your Hakeli, Lanciaris, Polyphylus, and Fulvus. So almost 90% of Chrysopogon species I have shown, and by looking at spikelet, you can identify them without any problem. I want to tell you that grasses are not difficult. So Vetiver, once upon a time, it was recognized as a genus Vetiver, but now it is merged with Chrysopogon. Okay, it has also lower spikelet and 2 pedicelled spikelet, 1 and 2 pedicelled spikelet, and this is a sessile spikelet. And thus, this is also now Chrysopogon. So it is a Chrysopogon lawsoni and Chrysopogon giganoides. See, Chrysopogon, I think verticillester inflorescence, 2 pedicelled spikelets, 1 sessile spikelet. I hope you will be able to remember characters of Chrysopogon. So this is also Chrysopogon. See this Chrysopogon, right? And Quoix, another genus which have about 3-4 species. This you can see this, right? Male and female flowers in different parts of the same inflorescence and fruit case closed, formed by leaf sheath. This case, which is formed around female spikelet, is a leaf sheath, not a glume. It is a leaf sheath. These are male spikelet. So we have Quoix gigantea. Then we have, you can see the diversity of these nut-like structures. Okay, so much diversity we get in size, shape, and color of the spikelet. Now, Eleonoras. See this Eleonoras genus. See this lower glume, which is having bulbous-based hairs, and on either side there are a bunch of hairs, and the lower glume is biforked at apex. Okay, biforked at apex, and that way, this is a grass which grows in extremely dry regions, in rocky places. In Aurangabad region, I have collected, and Eleonoras having about only 1 species in India. If you know this, that means you know Eleonoras in India. Euclasta, this is another genus with a single raceme, and you can see these spikelets. Okay, so I have given characters in bold. You read it otherwise it will take a lot of time for me. Already it is 11:56. I don't know what to do. Elopsis. Okay, so see this Lasiurus, which is found in Rajasthan area, and it is a boon to the animals of Rajasthan. Okay, then this is the Lasiurus. This is, see, Pogonacne racemosa. You cannot go wrong. See this on lower glume, the upper glume with a thick tuft of hairs from the middle. This tuft of hairs, if you remember, this is a Pogonacne. It has only 1 species, Pogonacne racemosa, and it is monotypic endemic grass genus of Western Ghats. Sahima, this is another very important fodder grass of our country. Then this is a Sahima, you can see the spikelets. Then we have Pondiopogon, Rhizopus, you can see the hairs. Then this is a Theliopogon, again it is a monotypic endemic genus, elegans. And see this crocodile-like lower glume of the spikelet, crocodile-like lower glume of the spikelet. I think you will enjoy them, right? And then Triplopogon ramosissimus is another unique plant, which is also monotypic and endemic to Western Ghats. Okay, Tripsacum, again it is a medi, and you can see this lower glume, right? Pistillate spikelet embedded in the internode, and the nodes truncate with central peg. So this is the way. Tripsacum. So it looks like this. This Tripsacum. See this upper male flowers, lower female flowers, and you can see these female flowers and these male flowers. Who will go wrong in identification? Nobody can make mistake. Maize is known to you, right? Then Rotboli is another very interesting group, subtribe. You see this, Irimo, Cloa, Muricata. See this, lower glume, right? Lower glume is so unique. I don't think you will need anything for identification, right? Glyphocloa. Then see this, Glyphocloa, various lower glume only I am showing, and you can identify all the species of Glyphocloa, right? Beautiful ornamentation, lower glume. Then we have Nesithia, which is also closely related to Glyphocloa, Hakelocloa granulis or Nesithia granulis. So you can see this, the lower glume is very unique. You can identify how it looks. See this lower glume, right? Then this is Manissuris myros. See the lower glume, the shape of the bloom. I hope you are enjoying these grasses, Manissuris, Nesithia clerki, right? This is Nesithia welcampi, which we described. Rotbolia, you see this Rotbolia, okay? It is a single spike. And Rotbolia, then your Hamarthria, then your Aphuras, all this tribe is very unique. Aphuras, then Dimeria, and Ichimam. Hello, Hamid. Hamid? Ichimam. Now, genus Ichimam is another important genus, which is, widely distributed throughout India, and it has maximum species in Western Ghats. It is, it is probably originated in India, and it is actively evolving in Western Ghats. And this Ichimam, you see, it has two spikes, and if you open, you will get two spikes, and the lower gloom of this Ichimam is of diagnostic value. Okay, you can identify Ichimams by looking at a lower spikelet gloom, lower gloom. You see this gloom. So Ichimam mangluricum, Ichimam tumidum, Ichimam rhizide, Ichimam boli, Ichimam impressum, Ichimam rugosum, Ichimam santapai, Ichimam species. Okay, now it is named after me, Yadavi. So you can see these Ichimam species which are we can identify. Now Clystacne. Now see this Clystacne, which is closely related to our this one sorghum. Clystacne, sorghoides, and see this lower gloom is covered with lot of coriaceous, thick, ovate, dark brown, densely hairy. So it is densely hairy. I have shown these characters. This is a sorghum. All of you know. I will not go. Saccharum. All these are ornamental grasses which are grown in gardens. Okay, Saccharum. But this is Eulalia trispicata. Okay, Eulalia trispicata. And one species is named after me, Eulalia srirangi. This is Eulalia srirangi. Right? Now, Imperata. This group of Saccharum is unique in having hairs. You see these plumose hairs. Imperata cylindrica has a 10 species, and it is a noxious weed in many places. Once it enters, it occupies entire grassland, and that way it is one of the weeds, and it has got plumose hairs for dispersal of these small spikelets. So if you see, now you see Saccharum spontaneum. I think all of you can identify this grass. You must have seen also. I need not to explain. Saccharum erianthus. Okay. Then this is Apocopsis. Now Apocopsis, just Apocopsis manglorensis. See this transversely cut lower gloom. It is a transversely cut lower gloom. This is a lower gloom, and you can see this lower gloom is transversely cut, and there is a single spike, and this is where you can identify Apocopsis. There are about 16 species. Then this is Lophopogon. Lophopogon, you see this Lophopogon, single head like in Florence, and these are the awns. You can see these awns. This is a close-up, and if you see the lower gloom, it is with 3 teeth. 1, 2, 3 teeth. So you look at this lower gloom and remember that in Tripogon we have tridentate lower gloom, and that way Lophopogon tridentatus you can identify. There are 3 species distributed in India and Australia. Now this is Lophopetalum from south. I have taken this photograph, and Lophopetalum, you see, I am yet to identify, but it is also a very unique grass, right? I am yet to identify its species. Right. Now let us go to Bhaidia. This is a genus which is endemic to India. Genus is endemic, and there are 3 species of Bhaidia found in India. Now see here, this is a sessile spikelet. This is a pedicelled spikelet. And one side of the pedicelled spikelet, lower gloom, is winged. This is a wing you can see. And by this wing, you can identify genus Bhaidia. The wings are very broad. Here also, for lower gloom also, of lower spikelet, the wings are very broad. And that way you can identify. Cymbopogon, all Cymbopogon species, if you crush the leaf and smell, they will have aromatic smell. They are aromatic grasses. And you can see sessile spikelet and 2 pedicelled spikelet on either side. So this way you can identify. Heteropogon. Now this heteropogon, see this heteropogon spikelets, stamens, right? This group is very unique. This is Heteropogon contractus, Heteropogon ritchii, and Heteropogon tritius. There are 12 species and racemes. These are the racemes, single raceme at the end of the branch you get, and then we have this horned lemma. Now, this is very unique grass, Isilima. I always tell one story. Isilima is I compare this spikelet. This is a spikelet, Ichimum prostratum and Ichimum laxum. This is a spikelet, single, not single spikelet, it is a unit of spikelets in which we have 4 pedicelled spikelet. This is 1, 2, 3, and another side 4th pedicelled spikelet. And these are again 2 pedicelled spikelet which are longer. You see this pedicel, long pedicel. So there are 6 pedicelled spikelets which I compare with Pandava. So there were 6 Pandavas. Okay. So 6 Pandavas, and in the center, there is a single female spikelet surrounded by 6 pedicelled spikelets. So I call it as a Draupadi. In the center, there is a Draupadi. All around, there are 6 Pandavas. And this way, nobody can make mistake in identification of genus Isilima. Remember, female spikelet is in the center, surrounded by 6 male spikelets, of which 4 are with short pedicel and 2 are with long pedicels, Nakul and Sahadev. Okay, so that way, Isilima is another genus which is a very important fodder grass. Okay, so you can see this. This is a female spikelet. These are the male spikelets. These are long male spikelets. These are female spikelet, and in the center there is a sessile spikelet. So this way you can identify genus Isilima, and in a Thymida and Isilima, only the spikelets here in Isilima they are whorled and they are one above other in Thymida. That way we can identify. They are closely related genera. Okay. Pseudo Dichanthium is a monotypic endemic genus of grasses in Western Ghats, having only 1 species. It looks like Heteropogon, but it is a different. Racemes are markedly curved, glooms glabrous, lower gloom of the sessile spikelet keeled and broadly winged and glabrous. So this is monotypic genus of Western Ghats. So this is a Thymida. Already I have shown you Thymida. This is Thymida pseudotrumila, Thymida quadrivalvis, Thymida triandra. So these are the species. Right? Thymida I have shown you. Now Capillipedium, then Batrocloa, I have not put photographs, just remember them. Dichanthium is another very, very important genus, and there are about 32 species in India, and all Dichanthium species are highly nutritious. They are the best fodder grasses of our country, and identification is little bit difficult, but if you sit with this, you can identify. Species identification is difficult. Dichanthium caricosum, Dichanthium armatum, these are the species. Then Pseudanthristia, this is another genus which is common in grasslands. Then this is a dichotomous. In Nagpur region, we have collected it. Dichotomous. See the lower gloom, see the upper gloom. Just like our Bhaidia, it has also very broad wing of lower gloom. So it is monotypic genus having only one species, that is Dichotomous fastigiata. Now this Andropogon is also common in throughout India. It has 122 species, of which 9 occur in India. Now, each, this one, terminal branch bifurcates and give rise to 2 racemes. This is 1, this is 2. 1, 2, 2 racemes. And they are just andropogony, spikelate, sessile spikelate and pedicelled spikelate, and sessile spikelate is female spikelate or bisexual spikelate. And if you break this from here, see this point here, from here, you get 3-toothed structure at the base. 3-toothed structure at the base. By that single character, 3 or 4-toothed type, you can identify genus Andropogon. So Andropogon pumilus is common in peninsular India. Okay. Then Syzygium paranjapianum is another grass which is also, which has, Syzygium has about 50 species, right? Then Hymonacne, see this Hymonacne acuigluma. See this single spike, raceme, pedicelled spikelate, and lower gloom is reduced. This is a lower gloom, which is reduced, lower gloom. And by looking at it, you can see. So this is a Oxonopus, which is grown as a lawn grass in our gardens. And then this is a Paspalum. Okay, this is a panicle of racemes. You can see these racemes. And these spikelates, they are on one side of the rachis. All spikelates are on one side of the rachis. That is Paspalum. Paspalum canary, variety canary, Paspalum scrobiculatum, Paspalum disticum. So these are the species. Then you see Echinocloa. Colonna is a common grass in all ponds and puddles along roadsides. It is a very common, and we have some 20 species, of which 7 occur in this one. See the outer gloom, which is reduced. I can give lecture for long time and explain all the characters, but because of our time limit, I cannot explain everything, right? Then this is a panicum, right? And this is a Sassiolepis. See this Sassiolepis, and the spikes, single spike raceme, and these are the pedicelled spikelates. Lower gloom is reduced. This is Digitaria. All Digitarias have glandular hairs on spikelates. These are glandular hairs on the spikelates we get. So Digitaria ciliaris is another major genus with many species. Now Trachis, I think Trachis you cannot forget. You see this Trachis and see the around the spikelates, there is a whorl of outgrowths, leaf-like outgrowths, and that way it is a very unique. Trachis muricata. There are 4 species from India only. All species have been described. Trachis muricata. This is a Trachis new species described. This is the key for identifying Trachis species. Now this is Acroceros, genus Acroceros, which has 15 species, and we have Acroceros munroianum. Now here, see this lower gloom, upper gloom, lemma, pallia. Okay. Now the spikelate is dorsally compressed or almost terate, glooms and lemmas with laterally compressed and thickened apices. So lemmas, these are the lemmas, these are the glooms. They at the tip, they are thickened. See this thickened tip. Okay. So only by looking at this thickened tip, you can identify it is a Acroceros. Okay. Acroceros munroianum. Remember this. Then this is a Alloteropsis simicina, which has lot of hairs like our Digitaria. Okay, but the inflorescence is a panicle, and that way we can identify Alloteropsis. Alloteropsis. See this, how it looks. Alloteropsis simicina. Now we come to Certococcum. Certococcum. See this spikelet. I told you shape of the spikelet. This is lower glume, upper glume, lemma, and pallia. Okay, you get, and see the shape of the spikelet. These are the forest grasses. They grow in the forest as undergrowth, and the spikelets are very unique, by which you can identify. There are 12 species, of which 7 occur in India. Then Aplis menus, Burmani. You can see this Aplis menus and see the on. Okay, glume has a on. Then this is also Pseudo-Echinoclova. Name is Pseudo-Echinoclova. It is a forest grass. See on spikelet, there is a bunch of hairs. When it matures, bunch of downwardly pointed hairs is developed, by which it get attached to the body, and then it is dispersed. This is Pseudo-Echinoclova polystachya. See this Pseudo-Echinoclova polystachya. Now here you can very easily, will you make mistake in identification with this photograph, and if you have lens, with lens only you can identify this grass, Pseudo-Echinolina, right? See this hairs, hooked hairs. Right. Now see this Cenchrus. Cenchrus means having lot of outgrowth. Now this Cenchrus has outgrowth which are very sharp, pungent, and inside there are spikelets, and there is a whorl of this involucre of spines or rigid bristles around. By this only you can identify Cenchrus. So this is the Cenchrus ciliaris. The this one, what we call the bristles are cilia-like, so ciliaris. Then you can see Cenchrus biflorus, Cenchrus ciliaris, Cenchrus setigerus, Cenchrus echinatus. You can identify all of them. Cenchrus and pennisetum. This is the pennisetum, and on this city we have a lot of plumose hairy outgrowths. So this is a pennisetum. Pennisetum purpureum, pedicelatum, and now Cenchrus is merged. Cenchrus and pennisetum is merged into same genus. Okay. So Cenchrus, this is the pennisetum. Now Panicum antidotale. Now this genus is one major headache for everyone. Identification of species is a very difficult task in Panicum. Okay, Panicum. Pseudo-Rhapis. This is again aquatic grass. Upper lemma is smooth. Bristles caducous solitary, formed by the prolongation of the branch beyond the terminal spikelet. So you can see this growth, and Pseudo-Rhapis piniscens. So this is a setaria. Setaria means city. Each spikelet is subtended by city. You can see this city, and then this is a spikelet with lower glume, upper glume, lemma, and pallia. So these are setaria characters, right? Spinotaprum. So in Kerala only I have collected this grass. This spikelet is embedded in the rachis. हा सर, मी तुम्हाला फोन करतो. मी जरा लेक्चर देतोय, फोन करतो. चालेल? चालेल. Spinotaprum. Okay, it is very interesting. See this. These are the spikelets. This is the rachis in which they are embedded. Now I think you can identify this grass very easily by this structure. It is an ornamental grass. Then spinifex. male and female plant. This is a male plant, this is a female plant, this is a female, you can see, and this ball rolls on ground, and Spinifex has only one species, Spinifex littoralis, male plant, female plant, and you can see this spikelets. Okay, so you can identify this. Then this is Eriocloa. In Eriocloa, very interesting thing has happened, that lower bloom, this is that lower bloom is reduced to a knob-like structure. So this is a reduced lower bloom. So that way we can identify genus Eriocloa. There are 30 species, of which 2 occur in India. Eriocloa procera and Eriocloa hathmensis. These are the 2 species which are found in India. Now this is Thuria. See this Thuria, and this is a terminal inflorescence with spikelets, and then it bends down, and the seeds are enclosed in this, caryopsis are enclosed in this. So bending down of this inflorescence itself indicates it is a Thuria, right? Thuria. Then this is a Eriocloa. Again, it is related to Panaceae members. See this hairy outgrowth at the base and spikelets, and lower bloom is highly reduced, Eriocloa. Then this is Milinus, Milinus rincellitrum. So this is a complex. This is a Milinus which has very, very small spikelets, which is common in now Western Ghats, Milinis. You can see this Milinis, how it looks. It has a great ornamental value also. So this is Milinis. This is the Milinis minutiflora. Milinis minutiflora. Lower bloom you see, upper bloom, lemma, palea, and that way it is a unique grass. Milinis, right? Then Morocloa. Okay, see these are the Morocloas, which was placed in a brachiaria, but now separated into separate genus Morocloa. This is Morocloa inciformis. Then Tricolina. See this spikelets are so unique. See these lower glooms, 2 lower glooms, then upper glooms which are on, and then lemma and palea. So Tricolina is another this. So Phayu 34, we have to go to 711 genera. So now Danthanoideae. In Chloroideae, we have Halophyrum, monotypic genus, which is found along seashores or in Rajasthan desert. You can see this grass, very robust grass, very important grass for desert area. And it looks like this. You can see this spikelet, single spikelet, right? Single spikelet, and see bunch of hairs at the base. So you can identify by this monotypic grass genus. Florets conspicuously bearded from the callus. This is a callus. Okay. So Halophyrum mucronatum. This is a mucronate bloom. You can identify. Then this is Eriagrostis. Again, it is Indopoa paporculina. You see the spikelets. The spikelets in terminal solitary secund spikes on twisted, lemma emarginated out 2 to 4 lobed or toothed at the apex. So this way we can identify. This is Octocloa. In Delhi region, we have collected Octocloa compressa. Okay, in Rajasthan area also, we get Octocloa, monotypic genus, Octocloa compressa. Then Salentvallia, which is described from Salent Valley by Nair, again, Sri Kumar, Vijay Nair, they have described this. And this Salentvallia is a monotypic endemic grass genus from Western Ghats. We have described one more species, Salentvallia, from our region. This is Salentvallia chandavadensis. So these 2 species are there now in the genus. Okay, so these are comparative accounts. I will not go in details. Chloroideae. Now you see, Inni-pogon. Inni-pogon means, inni means many, pogon means awns. Now see this spikelet, which have many awns, 1, 2, 3, 4, 5, 6, many awns. So lemma, 9 nerve, the nerves produced 9 awns, lemma strongly compressed or rounded. And that way, this grass, which is a common grass in South India, in drier regions, it is a Innipogon elegans. It has 28 species, right? See, Innipogon, how it looks. Then we have Eriagrostis. This is very, anybody can identify because of compressed spikelets. They are compressed like cypressy. They look like cypressy spikelets. So this is a spikelet, and there are many lemma, lower gloom, upper gloom, then lemma, palea, lemma, palea, lemma, palea. So it is many flowered spikelet. And this one, you know, is Eriagrostis. All of you can identify genus Eriagrostis. Then we have Chloridae, Zosia. It is a cultivated grass, jumping grass we call, and you see the spikelets. Spikelets are very small, and see the spikelet. Blooms of the spikelet smooth, compressed, shining. They are shining. Spikelet arranged in rigid, erect spikes. They are arranged in rigid, erect spikes. This is Zosia matrila. This is Crypsis. In South India, we don't have, but in colder climate, it grows. This is a Crypsis. You can see this inflorescence, Crypsis inflorescence, right? So inflorescence spikate, very dense, subtended by the inflated sheath, right? Bearing a rudimentary blade. So that way we can identify. This is Sporobolus, is another very unique genus, Sporobolus capillaris, and this Sporobolus is a unique in the sense you can see this Sporobolus. Now all around there is a pericarp. This pericarp, when the grains are made wet, the pericarp comes at the base and the seed or caryopsis comes out. So grains usually with the pre-pericarp we get in Sporobolus. This is the only genus which is unique in its having grains with a pre-pericarp, right? Now let us go for Aleropus. Now this is Aleropus lagapoidus. See the terminal head-like inflorescences, and this is a spikelet, right? Then this is Pomerula. In South India, we have Pomerula in an extremely calcareous soil. In waterlogged conditions, we get this grass, but it's a very unique grass. You see this unique grass which grows like this. It is a monotypic Pomerula cornucopia, right? So see this spikelet. So it has Pomerula, many whorl of bract-like structures, and in the center spikelet is there. So this is that Pomerula. It is monotypic, and you should not make mistake in identification. Dactyloptenium, I think all of you can identify. On one side we get the spikelets, and you see this. This rachis has elongated. Dactyloptenium, we have 13 species, of which 4 occur in India. Dactyloptenium syndicum. Then this is Tragus roxburghi, or it is also was known as Tragus biflorus. Tragus roxburghi, which has a, you see on blooms, we have hooked hairs. So you can identify this in all dry regions. Anywhere you go, dry places, you will get this grass. This is the Parotis indica. You see this Parotis indica. Only looking at spike, you can identify this Parotis. And this Parotis indica is common in South India. And the entire field, you can see this Parotis panorama, how these spikes have come and spikelets we have seen. So it looks like this, Parotis indica, right? Now, Parotis, so I have shown this. And this is Parotis. Initially, it was placed in Lopholepis ornithocephala. Now it is, it is transferred, or it is treated as a species of Parotis by Kellogg. So Lopholepis ornithocephala, you can say, or Paratis ornithocephala. Now see this raceme. This is a pedicel. Then this is a spikelet. The gloom is modified in such a way that it looks like bird's head. So it is known as Lopholepis ornithocephala. Okay, Lopholepis ornithocephala. You will enjoy it as a ornithocephala, right? You can identify. Then it is Leptothium. It grows in Rajasthan, extremely dry, sandy soil. And see, it looks, these are sessile spikelet, pedicellus spikelet, and it is also near to our what is that? First genus, Paratis. And see this ornamentation. You can identify. It is a very small grass. Then this is Acracne racemosa, which is close relative of our this one. What is that? Curacana, Elusine curacana. This is Chloris. By looking at it, this one, you can identify Chloris. Cynodon is another genus with us. Then this is a Dinebra, which retroplexa. Dinebra retroplexa, it has only 3 species, of which only 1 occurs in India. Dinebra retroplexa. Then this is a Diplacne. It is a monotypic grass genus. It has only 1 species, Diplacne fusca. You can see these spikelets, right? Leptocloa, another genus which has 40 species, of which 5 occur in India. They are closely related. You have to make little studies. Our photographs will be useful for identification. Lepturus, this is a grass which we collected from Karnataka, and see these spikelets are embedded into rachis of the spikelet. Okay, they are embedded. Then Microcloa is another genus, Microcloa. You see this single spike, Microcloa, one-sided spikelets. So one-sided spikelet, Microcloa indica. Then Sconfidia. This is another genus, Sconfidia gracilis. Tetrapogon, again it was kept in Chloris, but now it is separated into separate genus, Tetrapogon, and you can see this on and spikelet. The shape of spikelet itself is sufficient for identification of Tetrapogon. This is a Silacryum. See the seed. Seed is very unique in grasses. It is ornamented and it looks like this. Grass looks like this. It is from South India. Then this is Elusine curacana, which is well known to you. Leptocloa I have shown. Now we come to Chloridoideae. Now this is Desmostychium. Again, it is a monotypic bipinnata. In Delhi region, it is a grass. Spikelets are very small. You see the inflorescence and you will be able to identify monotypic grass genus. What is Desmostychia bipinnata? So Iriagrostiella, again it is a like Iriagrostis. You can see this spikelets are in 2 rows on the rachis. So you can see this rachis, Iriagrostiella biparia. Now this is another grass, Melanocencris, which is unique. You can see these spikelets are in bunch. Manuceris jacquimonti, Manuceris jacquimonti. So I will run through slides. Time is over. So this is Melanocencris. Then Oropitium, Thomium, it is a small grass. Tripogon is another important grass. And now Colacne, it looks like this, Colacne, right? Colacne simplicicula. Then this is a Colacne minuta, which is endemic to India. And Hubbardea heptanuron. Already I have told a story last time, so I need not to go in this one. Hubbardea heptanuron. Isacne, you can see this Isacne spikelet, you can identify. So there are 29 species which are found in India, right? This is Limnopoa, which is found only in Kerala. Okay, Limnopoa. Limnopoa, only found in Kerala, monotypic endemic grass. Then we have Arundo, it is a large grass, Arundo donax. Then we have Arundo, this is the looks, you see the hairs, which are very unique on the rachis. That way we can identify. Then this is Elytroporus spicatus. In drying rice fields, you can get this grass. It is also a very unique grass, and you can very easily identify by looking at inflorescence. Then this is the Phragmites, which is again common grass. And in river beds, we get this Phragmites. And you can see this panorama of Phragmites kirka. And we have Phragmites kirka, Saccharum officinarum. So these are the grasslands which provide habitat for many animals, birds, and so on. Okay, Jankeria is one grass which is found in South India on at high altitude. Okay, then Danthonidium is one which is very unique. You can see these spikelets. It grows on. This is a lateritic rock. On these rocks, we get this grass, and you can see this spikelet. We can very easily identify by looking at spikelet. Now, I have taken some 5-6 minutes more, but really speaking, grasses, my students are sitting in grasses, and in India, P.J. Nair was a very important grass specialist. Unfortunately, we have lost him in this year. Now, another thing that who will study these grasses? They are unique, they are interesting. They play very important role to maintain this Mother Earth. It is most important biological resource of human being. Yet we run away from grasses. Now these grasses only provide you food, meat, milk, ice cream, sugar, and whatnot. Even bamboos. Right from our birth, bamboo comes as our cradle, and when we go on bamboos only, we go to our smashan bhumi, criminal also. So grasses are related to our life greatly, right from birth to the death. And therefore, dear teachers and students, let us study grasses and make it lively subject. Thank you very much for patient listening.
Participants, already chat box is availed. You can chat and ask if some questions are there. Please, if you have any queries, please put questions. Anyhow, sir always will be motivating, and sir will be always giving so much inspiration for us. And already he has made his botanical garden and many more activities he is doing. Recently, he was found in the Indian Botanical Society meeting also. He is a celebrity for our country, who is really working wonderful for the conservation, for the characterization, and whatnot. So many activities sir is doing from long time. I think more than 4 decades he is putting his time for all the systematics. It is a rare opportunity sir is available with us, though he was very busy in all his assignments. He gave time for us, and really his lead garden in the Shivaji University is a proof that he is really a gem of our country.
Hello, students. Not students, my teacher friends. Hello, somebody should talk to me.
Talking, I have made a mute, but I
some of them can unmute, no, so that they can express themselves.
I have made unmute.
These grasses I want to make popular in our country so that we can utilize them for our betterment. They should understand the importance of grasses, because it is a very, very, very important source, biosource for many purposes. Okay, and that's why I know in 1 hour I cannot teach grasses to anyone, but just glimpses I have given. And really, if we want to identify grasses, it needs 3-4 days training program so that they go with confidence and they identify almost all genera. There are 266 genera, and in 1 hour I cannot complete, because one genus at least takes 5 minutes to explain. And with that rate, you can understand how much time it will take. Anyway, I have just introduced. Those who are interested in knowing grasses, please contact me, and whatever help you need from your area, try to collect grasses, try to identify them. If you cannot identify, just send me one inflorescence. I will let you name of the grass, and that way you increase your knowledge of grasses. Thank you very much.
Sir, there is a question, sir, actually. How to identify, I can't pronounce it properly.
Chivonacne and quoits.
Yeah.
Right? In Chivonacne, the cell of the female flower, okay, shell, round structure, is from gloom. Okay, is from gloom. And in quoits, that cell-like structure is from leaf sheath. That is a major generic difference between Chivonacne and quoits. Okay, so in Chivonacne, you will not find lower bloom inside, spikelet, but in quoits you will get lower bloom also. Outer cell-like structure is of leaf petiole modified into cell-like structure. Is it okay?
Yes, sir. Thank you, sir. Thank you for such a very keen on the grasses, and not only grasses, but lot you have taught us in all the aspects of plant diversity, characterization, your talk is so full of pictures, and you have explained so well for us. Thank you so much, sir. Thank you. Thank you.
Thank you. Thank you so much, Amit Saab. Let us now. We will meet someday.
In front of everybody, I'll ask is, sir, again in the month of June or July, so we will have one more online meeting where exclusively we'll be doing the only 100%. About grasses only we'll be doing. So you invite your students, scholars who work with you also, so that we can have exclusively one lecture workshop of 3 days on 100% this concepts only. As you told, you have cut down the your presentation to the time constraint. So in future, I will be requesting you again back for applying Academies program again on this concept.
Okay, sir. No problem. It is my pleasure. It is my pleasure.
With your permission, I would like to end the meeting now.
I will also leave them.
There is one more question has come. Can you see the chat box, sir?
Sir, in case of Eragrostis iniloides, I found glands on the inflorescence, but it's a divide from glands, wrote in Bohr's book. Now in Eragrostis, on the rachis, we get some glands. There was some good article also on presence of glands. What is their function? They secrete some, and ants visit that. But your question is, in case of Eragrostis iniloides, I found glands on the inflorescence, but it's a divide from glands, wrote in Bohr's book. Okay. So, Bohr, that time when it is he has written his book based on herbarium. Probably he might have missed to see those glands. Okay. So, but he has written such a wonderful Bible for all of us, and some characters have been missed. Now your people to find out more and more character and make it more knowledgeable in grasses. Is it okay? Jalandhar. So Okay, sir. I will leave now.
Sure. Thank you, sir. Thank you so much.
Okay.
Dear teachers, we will be back again at 1:30. Thank you so much. Have a